A beautiful silhouette in the clouds over Bunso in the eastern region of Ghana. This does not happen in daytime. Thus, at this time, the unique identity of the catchment area is dead. Soon, Bunso will catch daylight to reveal its features. Significant among these are tree shrubs and plantations in the wild. Wherever natural vegetations are to be found in Ghana, they are under threat from human activities. In the exigencies of development, trees are mowed to make way for construction, bush burning, mining, farmlands, and any other human-related activity. In the process, all kinds of plant species are destroyed. This is where the CSIR's Plant Genetic Resources Research Institute comes into the picture with 1 foot ahead of destruction. The PGRRI, as it is called, serves as a bank for certain species of plants regarded as sacrosanct by scientists. The institute conserves plant genetic materials for future uses, and it indicates some of these materials have been preserved since the 1970s. Beyond the basic plant culture of photosynthesis, which scientists have taught through the ages, the CSIR scientists of Ghana tenaciously walk the distance of plant lives. Plasm, the genetic material of germ cells, is of importance to the plant bank. Seeds that contain moisture are dried in sunlight, in the oven, or the placement of silica gel near seed samples. The gel emits heat and must not touch the seed bag to scorch and destroy it. Recalcitrant seeds are the ones without moisture and fails in low temperatures. These cannot be preserved and so are taken to the field for planting.
We do our purity test. The purity by removal of inert materials, stones, debris, broken ones or infested ones to get a clean seed.
The plant bank of PGRRI subjects seed samples to germination tests to avoid preservation of sterile seeds.
This will help us to know that when we're putting the material into storage, it was having a germination percent of about 85 or 90. So in 2 or 3 years, if we bring the materials out, we'll test again. If it falls below the 85, then we must send it back to the field for regeneration.
The seeds to be placed in cold room are tested for moisture contents, and beyond certain thresholds, seeds could sprout inside the bags, neutralizing the original intentions. Another layer of seed quality control is taking the fruit that contains seeds to the lab to check for diseases. This is turkey berries called Yasantua by a section of Akans in Ghana. The turkey berry, generally resistant to diseases that afflict vegetables, is pictured here with blackened spots, considered an infection.
And normally, if you want to go about isolation, you take the tissue that is dead, normally called the necrotic tissues. You take the necrotic tissues and you don't take it from the middle, but you take it from the periphery. And that is the boundary between the healthy and then the diseased tissue, so that you avoid contaminants, the other organisms that may come in to take advantage of the decay portion, because they are normally saprophytic and they depend on dead organic matter.
Infected fruits or stem cells are isolated from conservation. The scientists are interested to know more about diseases before isolation. And one of these procedures is the example of a tissue from an oil palm tree with disease symptoms.
What you can see there is a tissue of oil palm, and the leaves and then the palm fronds were infected with some disease symptoms. And so we took samples of the disease parts.
The causal agent is magnified in an incubator machine. Another phase of plant conservation by the CSIR's Plant Genetic Resources Research Institute is tissue culture. It simply refers to growing plants under artificial conditions.
The cultures are started.
Plant parts are placed in tubes and kept under low temperatures or in air-conditioned rooms. Yam tissue in her right hand and cassava in the other. Taro, potato, and many other plant tissues are also here.
This is backup for what is at the field. It gets moved to the field in the instance where what they have at the field gets lost and they can get lost due to like maybe disease outbreaks. They are conserving all right, but still there because the environments are not controlled. So this is like backup. So these are duplicates of what they have on the field.
Handlers of the plant bank at PGRRI at Bunso ensures that the tissues are properly cultured to avoid disease organisms becoming a part of contents of the tubes.
When you pick the material outside, you have to make sure the material is clean of these microorganisms, so that when you grow them, the microorganism doesn't compete for nutrients you produce, you give to the plant.
The Forestry Commission and its partners, some of which is the Council for Scientific and Industrial Research, CSIR, have been fencing plantation in certain parts of the country and maintaining them as a forest reserve. For the Council for Scientific and Industrial Research, it has been moving a step further in trying to harness certain particular species of plants for preservation. The Plant Genetic Resource Research Institute at Bunso has been serving as a plant bank or bank for germplasms. For the Animal Research Institute, it has a pasture museum here at Pukwase.
This is a very big national asset. And so, in fact, throughout the West African sub-region, this is the only pasture museum. The materials you see here, to the layman, they look like grasses, ordinary grasses. But these are materials that have been brought from all over the world, some from Brazil, some from Australia.
Those who raise or mutilate any plant, perhaps considered by them as useless, may be destroying something treasured by scientists. Here you are with animal fodder, classified for their biomass yields, animal acceptability, and nutritional quality. Any encroachment on CSIR lands might well erase time-honored academic research. Napoleon Atukitu reporting.